Well, good morning. Thank you for having me here this morning. Like Nancy said, I'm Ashley Bales and I'm the collections manager of the BRIT Herbarium. And I'm excited to talk to you more about the herbarium today and how we're preserving nature's legacy, not just at BRIT, but at the Fort Worth Botanic Garden. So I've already gotten my introduction a little bit, but I think growing up I always knew that as a career I wanted to help people. And I thought at first that meant, pursuing medicine and helping people that way, but I think all along I knew that my way to help people was going to be through plants. So what does it mean to preserve nature's legacy? Does anyone have any ideas they want to throw out there? Do you think you all preserve nature's legacy? Yeah. Yeah, seed saving, absolutely. Anyone else?
No pesticides.
No pesticides? Absolutely, that's a good one. So I think that the role y'all play in your garden club is similar in a way of preserving nature's legacy. And so I want to share a little bit about how we're doing that at BRIT and at the garden. Has anyone been to the Fort Worth Botanic Garden before? Okay, I'm glad most people have. Well, so this is a map of the garden, and in the red circle you will see where the BRIT building is. So it's this little L-shaped building when you come in through the entrance off University. It's on your right-hand side. It's the building with the living roof. You'll see plants on the very top of it, and also has solar panels. So that is where the BRIT Herbarium is, and all the research and our education classrooms are. We also have a gallery, an art gallery in this building. So next time you visit the garden, definitely come over to the BRIT building. There's more to see over there. So if you weren't aware already, in 2020, the garden and BRIT actually merged to become one institution. So before 2020, the garden the city of Fort Worth managed the garden. And y'all know it's the oldest botanic garden in Texas. And BRIT was moved to the building on this campus in 2011. So for about 9 years, we were neighbors, and then in 2020, we decided to be the managing partner of the garden, and it's only gotten better ever since, in my opinion. So our mission now is to explore the critically important world of plants, to collaborate how they play a role in our cultural and natural environments, and it's how we engage people to conserve nature and improve our human experience. So we do that through our education programs, our adult education programs, and we have researchers coming to visit the garden using our herbarium collections. So how do we preserve nature's legacy? We do that at BRIT in a few ways, through our herbarium, through our library and archives, our BRIT Press, and our Texas Plant Conservation Program, and then also just our general research botanists working on taxonomy and evolution of plants. So you'll see a few pictures up there. Does anyone know what a herbarium is? You don't count. Has anyone ever visited a herbarium maybe besides the BRIT Herbarium? Yeah? Okay, so we've got BRIT Herbarium folks. There are over 3,000 herbaria worldwide and about 700 in the United States. But this is not a new concept. Herbaria have been created for hundreds of years. So the oldest herbarium that we know of was in late 1400s, early 1500s. So in this bound book were dried plant specimens, and in Latin was inscribed in tibi pertuis redentem floribus hortum, which means here for you a smiling garden of everlasting flowers. And they really do last a very long time in the right conditions. So this is what kind of a modern herbarium looks like, not in bound books, but single sheets filed in sort of a way that a library is filed. So I can go into our herbarium, go to a specific plant family, and I can see specimens from Texas and then all around the world, if there are plants all around the world. So the way it's categorized is taxonomically, by family, and then by geography. So we have different colored folders that you might see in this cabinet right here, and each colored folder represents a different part of the world. And we have specimens that touch every continent on the planet, which is really exciting. So we're the 9th largest herbarium in the United States at about 1.5 million specimens. And this isn't just the vascular plants that y'all are kind of seeing on your tables right now, but we also have algae, fungi, lichens, bryophytes. We have a wood collection, which is called a xylarium. We also have a pollen collection, or a palynological collection. And like I said, touches every continent on the globe. We recently found 2 mosses from Antarctica, so that solidified it just this year. So we didn't even know we had specimens from Antarctica until this year. And our oldest specimen was collected in 1791. So that's the image that you're seeing up here on the screen. And the only thing the label tells us is that it was collected in Mexico. Well, if we think about history and think about 1791, Mexico and Texas were kind of one and the same, right? So it's possible that maybe this specimen was actually collected in Texas and not Mexico, but we're not sure. So what are these specimens telling us? So each of these sheets, and these are just laminated images, so they're not the real deal. But what you're seeing is the plant itself along with a label, typically in the bottom right-hand corner, sometimes in the bottom left. And this tells you everything about that point and place in time about that specimen. So who collected it, when they collected it, where they collected it, what the plant is, and then anything else about, general information about the habitat, maybe more of a description about the plant as it was collected, like flower color, or maybe the scent, maybe if there were any insects around the plant. So we can get a lot of information just from this label, and it's really important data. So just right here, I'm looking at a Texas bluebonnet, all from Tarrant County, all collected in May, but one was collected in 1909, one was collected in 1964, and then the other one I think in 2023 about. So I can use this information, look at one species in one county, see what has changed over time. Are they still blooming around the same time? And according to these specimens, they're all in flower in May, so that's looking good. But this information could be useful for researchers to see maybe has climate change affected some of these distributions of plant populations? Are they moving more north? Are they blooming any earlier? So this information from these labels can help us understand a little bit more about these plants. So I want y'all to look at the sheets on your table, and I want to hear from you maybe what's something interesting that you might see from the label. Some people like whenever they find a specimen that was collected on their birthday. I know I have before. You've got one? Oh my gosh! Okay, we've got one birthday plant over there. Anyone else have like a question or something that they want to point out on one of these sheets on the table? And a volunteer actually put together some of these. You might see a piece of paper on the back with just some more facts. So a volunteer put that together for us a few years ago.
That's what I wanted to comment.
Yeah.
That one from Mexico.
Uh-huh. Thaddeus Hankey?
On the back.
Uh-huh.
What the common name is.
Uh-huh. Is it wrong?
I don't know.
Oh.
Octopus bush.
Uh-huh.
And it says it's used for fish poisoning.
Oh my gosh. Well, that's something new. I'll have to point that out next time I talk about the oldest specimen, because that is our oldest specimen right there.
My feedback would be please put the common names on
Yeah. Yeah, because I mean, I know I speak in scientific names, but I know most people really refer to plants by their common names, so I'm still learning common names. Does anyone have anything they want to point out about their specimen that they might have on the table? Yeah, what do you have?
Well, it's a sheet of something that says it's carnivorous.
Yes. How can it be carnivorous? Yes, so this is an interesting group of plants, carnivorous plants, and you'll notice how tiny those little things are. They're aquatic, so what they can do is sit on top of the water. They have these little pods, and it really sticks and traps these insects and bring them in. So super tiny insects, but they are carnivorous, Okay, well feel free to keep looking at the sheets. Yeah, what do you have?
This is kind of interesting. It's called the shooting star.
Shooting star?
It says from Illinois. It says it's of the primrose family.
Uh-huh.
It usually blooms white, but it appears lavender in the northern areas
Interesting, okay.
with the colder temperatures.
Oh, I think there could be several environmental factors or other things that could affect maybe how that the petals present in a certain color. Absolutely. So Illinois, okay. Wonderful. Yeah, continue to, you can just spit things out if you find something. So are any of you curious about how these sheets got on the table in the first place? How do we have laminated sheets of these specimens? It's a long journey. It is a long journey from these specimens to be out in the field where they're growing to eventually on the table where they are here right now. A collector, a botanist, will go out into the field and they will collect specimens either based on a specific, grant that they have. They might be interested in a certain species that they are destined to go out and find, but they will take the plant, they will press it in between sheets of newspaper, press them flat, and I have a field press over there that I can show y'all in just a second. But these plants, not just the plant is collected, but also data about the plant. So all the data you're seeing on the label, that data is collected at the time the plant is pulled out of the ground. There's also folks, maybe that collector or a team who's taking images of these plants when they're out in the wild, because I'm sure you can imagine once a plant is picked up, pressed, flat, and dried, it loses a lot of its characteristics. Either the colors will fade over time or some of those other characteristics that we might not know of if they didn't write notes about it. Once these are pressed and dried, we usually receive them in the herbarium. They get frozen at -40 degrees before they step foot into the herbarium. And then on our end, after the collector has given us these specimens, it's our job to mount them on archival sheets of paper. Then we digitize them, so imaging them and then transcribing the data that you see on the label into a database so people can actually find these images. And then finally, we will file them away into their cabinet in their correct place so that we can always go back and reference them and pull them out if we need to. So behind the scenes, once these specimens are here, this is kind of what it looks like if somebody is mounting a plant specimen. So you'll see there is some newspaper off on the side. So a volunteer pulled that plant material out and they're arranging it on the sheet in order, because once it's glued down to the sheet, that's it. So there is a little bit of training involved because we want the way it glued to be able to be useful for researchers later on. So we make sure that certain characteristics are showing or are facing up or down. So it's a little bit easier for researchers to continue using these collections and referencing them. So about on average, we mount about 750 specimens a month. And then imaging, we are completely imaged all of our 1.5 million specimens, but we're continuing to grow. So we are still imaging over 1,500 specimens a month with volunteer help. So to preserve these, it's not just physically, but also digitally. So we have an online database that anybody around the world is able to use. They can search our collections and other herbaria's collections that host their specimens on this website. We recently finished up a grant from the National Science Foundation to digitize all of our specimens from Texas and Oklahoma. So we have digitized all of our specimens from Texas, and we're working on what's called georeferencing. So taking the locality data that you see on the label and putting a point on a map, a latitude and longitude, and some degree of uncertainty about where it was collected. So it's nicer these days, people use GPS and they put coordinates on their labels, but for a lot of these specimens, we don't have that information. So using our protocols and best practices to get a good point on a map. And again, this helps with researchers for, again, distributions of plants or even finding new populations based on some of the habitat data that could be seen on these labels. And then, like I mentioned, everything goes through a big freezer at minus 40 degrees for several days. And that makes sure that we don't have any mold or pest insects that end up coming to the herbarium, because you can imagine these are probably very tasty for certain insects. And it's also a very flammable collection. So these cabinets are sealed tight, and we have a relatively low humidity and temperature in the herbarium, because we want these to last as long as possible. Our oldest specimen from 1791 wouldn't be in that great of shape if it wasn't kept in the great conditions that we have these specimens in. So I want to move on to our botanical library. And if you don't know kind of the background of BRIT and how it came to be, it all started with the Southern Methodist University wanting to give up this herbarium collection in the botanical library. And there were a few researchers who said, oh heck no, this is too valuable of a collection to just go in the trash. So they picked up the collection of herbarium specimens and botanical books and brought it to Fort Worth in 1987. So that's how BRIT was formed. So we do still have our botanical library, over 125,000 volumes. And this is specifically a botanical library, so all books that you can even imagine about gardening and plants and all the things, it's just wonderful, even botanical art books. And the botanical library also has artwork that has been donated over time, because I'm sure you guys can appreciate the connection between science and art and all of the things all within our library. Our library is non-circulating, but it is open to the public, and you're always welcome to come in and use some of the resources in there. And it's not just the books, but also archives. And there's a big connection with these specimens that you see on the table, and the archives and field journals and photographs that have been taken by these botanists and kept over time. So one of the big projects that the BRIT Library is working on is the Sherwin-Carlquist project. They're working with the California Botanic Garden. The California Botanic Garden Herbarium holds Sherwin-Carlquist's herbarium specimens, and our library has all of his field logs, photographs, and slide images. So this project is to digitize both of these collections physically that are separate and unite them with their data so that you can look at one specimen, you can see his field logs, you can see all of his images that he took from decades ago, and it really paints kind of a bigger story for just one, moment in time for this person. And he wasn't just a botanist, but he has all sorts of different images from all over the world that just really, again, kind of give you a piece of history from him himself. So our BRIT Press, I have a lot of books over here on the table I hope you can kind of swing by and look at. We have been publishing books for many decades. One of the big ones, maybe you've heard of it, the Illustrated Flora of North Central Texas. So any plants that you ever want to know about in our area, you would know what it is based on this book over here. And it's heavy. There's a lot of plants in North Central Texas. We also have the Trans-Pecos Flora, and then we also have East Texas Flora Volume 1 and are currently working on Volume 2 of East Texas. There are a lot of plants out there. Also the ferns and lycophytes of Texas. Did you guys know that our state has the most ferns native to any state? Yes, and a lot of those are out in the Trans-Pecos area, which I think a lot of people are surprised by too, given that it's so dry out there. You would think East Texas would have more, but really with such the unique elevation and geology out there, gives rise to all of these unique ferns out there. And so inside one of these books, you'll see, in some of them, we have an illustrated picture of the plant itself, and then for each species, a description and how to identify that plant compared to others that are very similar or closely related. And then also we publish twice a year a journal of publications called JBRIT. It used to be called CIDA. And this is a way for us to help researchers get their research out with these publications. So in one of these journals, you may see new species being published, new species being discovered. And a lot of this work comes from researchers who have dove into the archives, the herbarium specimens. They're looking at all of this to come to these these conclusions. So it's a wonderful place to just see what's new with the botanists these days, and also just ecology, evolution, all of the things can be found in these journals. So really at BRIT, we kind of have this cycle of knowledge. You know, we have people going out, doing the field work, depositing these specimens in our herbarium. We're making these data and images available to the public, to researchers. Those researchers are able to study the specimens, look again into the archives, into literature, publish their findings, and then we can kind of take action on what the findings are and see what we can build next. So our Texas conservation team does a little bit of this kind of in action. They have a goal to preserve our most rare and endangered species in Texas. There's a lot of them, so they're working one by one. And one unique plant that they've been working with is a quillwort called Eriocolon coronichianum. It's an interesting name. It is just this tiny, tiny thing that only grows in a very specific habitat in Texas, and also a few places in Oklahoma and Arkansas. We have a seed bank that we have been collecting these seeds of rare, threatened, endangered species, and just now our team has successfully been able to germinate these seeds and see that they're still viable. So what the methods are working, so this was really exciting just this year that they were able to do. But they've been able to also save populations whenever they've been in the midst of potentially being bulldozed by TxDOT or other construction going on. So the opportunity to protect the plants is a lot with this group. And so here's a little bit closer up of one of those seeds starting to germinate, and this is in their seed lab right here. And so there is this link, you know, between the conservation portion and the collections that we have. They would not have been able to rediscover some of these populations if not for looking back at historic records of these herbarium samples and seeing where they were located, and they're able to find new populations, again, based on potential unique habitat that they can notice on the data of these labels. And just our campus in general, I mean, you guys have been, most of you guys have visited the Fort Worth Botanic Garden, but I bet you didn't know how much research is really being done even just on our campus. We frequently have been doing prairie burns to see the resilience of these plants come back, excuse me, coming back year after year. We also, last year I believe, had goats in the garden to eat away at some of our kind of wooded area. Again, we want to get rid of some of the yucky stuff coming in and leaving room for more natives to pop up. So I think next spring the goats will be back, and also sheep. So if you're interested in seeing those animals, you can come to the garden. No horses yet. Back in the day there were horses. I heard it wasn't the best idea, so the smaller animals for now. Yeah, oh absolutely, they absolutely did. And you'll see a picture of a tiny, tiny mushroom in this picture right here. This is a new species that was discovered on a single tree in the Fort Worth Botanic Garden. So you may have heard there's news to create a family garden, and it's opening up next year. So this was in that area, but we made it a point to say we are preserving this tree, we can't knock it down. This is where a new species was discovered. So new species are being discovered at the garden, even though they're super, super small. And we are also doing more bat research in the garden. So the past 2 years, we've been doing monitoring to see what species might be using our garden as a home. And we have potentially 7 bat species in the garden, and 3 of which are species of greatest conservation need. So based on those findings, we're now moving forward and hoping to work with the bat sanctuary out in Mineral Wells. Maybe we can do some bat releases. And we're just hoping to educate the public more on bats. Just knowing that we're providing a really good habitat for them is just wonderful for me to hear. So you can be a part of preserving nature's legacy, and I think you guys already know that just based on the work that you guys do in the community, being an advocate for native plants, of course, and protecting our habitats, volunteering or giving your time or resources or just visiting the garden, joining us for one of our adult education classes. A herbarium specimen is a preserved moment in time, but its story continues to grow, and I hope that you'll take what you learned today and enjoy herbaria and just have a greater appreciation for the science that goes behind these specimens. Thank you all, and I hope I can give maybe a demonstration of how to search our online portal if there's time. We'll see. But I will open the floor for any questions anyone has.
Documenting those acts.
So we partnered with a Texas Master Naturalist Cross Timbers chapter. They were working with nature trackers, and they had audio moth devices that we set up in the garden that monitored and recorded sound for like 2 weeks at a time. They have some kind of program that they use to analyze that data, and so based on the echolocation sounds, they can tell the difference between different bat species. So that's how we know, and there's like likelihoods, you know, highly likely based on this echolocation or least likely improbable. So that's why I say up to 7 maybe, because a few were maybe potential or not as likely, but it's a possibility. Yeah.
Ashley, okay. Is it possible that one of the large data centers you could coordinate with around the world and collect all this data, somehow make it a big thing?
So our online portal is actually kind of a subset of other portals, and we also, our data gets transferred to a global biodiversity kind of database. So there's a few ways that people from around the world are able to access our collections, and we try to put them in as many places as possible for people to reach them. So there is a really, like a worldwide, where all of these herbaria's collections are kind of going into the same place. So I can also like bring that up if you're interested in seeing that. And it's not just herbarium specimens, it's other types of museum collections as well.
In 2011, you partnered with the Amon Carter and had an art exhibit.
Yeah.
And it was fabulous. I still remember it today.
Oh,
I just had to Google, though, the year. Have you thought about having another one, or are any of those in the works?
So I know that we are frequently, when, I'm trying to think of what there is a big thing where you can go from museum to museum, art gallery to art gallery. Is it gallery night, I think? So we usually participate in gallery night, and we do, frequently rotate, because our library has tons of botanical art that we're able to utilize and put into exhibits. I don't remember the last time we've partnered with the Amon specifically about doing something, but I mean, it's a possibility. So I only started with BRIT in 2017, but I'm happy to take it back and revive it if you want it back. We can see what we can do.
It was nice.
Oh, I love to hear that. Yes.
I volunteered in the herbarium 15 years ago.
Love it.
And one of the most fun things was each sample was wrapped in newspaper. We were doing old samples.
Yeah.
And it was really fun to see what was in the newspaper, what prices.
Yes.
What prices grocery stores were selling and what the news stories were.
That is one of my favorite things too. Just and we usually, I am not usually the person kind of doing all of the mounting of the specimens, but I encourage volunteers who are, if you see a newspaper that you like, keep it, and we can save this, because you're right, some of them are super old, depicting like really big moments in history too, so it's kind of interesting that newspaper was used to preserve a plant, but we're also like kind of preserving history in a way too with that, yeah.
I'm questioning why you use newspaper, like the lead in the paper. Does that not change the consistency of the plant, or why is that the best thing to use?
That's a good question. I think the material that the newspaper is made out of helps to dry the specimen a lot better. If you compare that to like magazine paper, something like that would probably not dry it out as much. So as far as I know, it's just what's been used. I mean, we have specimens, if y'all are familiar with the father of Texas botany, Ferdinand Lindheimer, he had his own press, and he would use his own newspaper and press his specimens in there. So it's just what's been done. I don't know any different, but I imagine that if we had an alternative, it would have to just be some kind of archival paper, cotton type of material, because I agree there could be something like, again, magazine paper that would kind of rub off, or we don't want anything kind of transferring to the plant itself. That's a good question. Yeah.
Who brings in most of the specimens?
That's a good question. So our botanists have received National Science Foundation grants over the past several years to go and collect in other countries, locally and around the world. So to give you a few examples, the one that wrapped up most recently was a Southern Philippines grant. So our botanists would go to the Southern Philippines. They went on 6 expeditions all around one island. And if you know the Philippines, there are thousands of islands, and it's historically been very understudied in this area, but it's definitely a biodiversity hotspot over there. And so that was the reasoning for wanting to go and go on these field trips to the Philippines. So our botanists collected vascular plants, bryophytes, and lichens, and they have already discovered new species based on those collections, which is really exciting. So besides kind of our funded projects, we do have local folks, local groups like master naturalists who may be, working in a specific area. They may be trying to restore like natural prairie, or maybe they want to document kind of certain land or someone's property. So we've gotten specimens donated to us that way as well. And then also students, there's not as many botany classes these days, which makes me sad, but there are a few local universities that do have some kind of field botany class, and so they will deposit their specimens. They're not the best, but we will take them. We might not take all of them, but those are just a few examples of some of the collections that we're still receiving. But herbaria also work kind of like a trading baseball cards kind of. So typically when a botanist goes out and collects a single specimen, it's not just that one, they'll likely collect duplicates. So if I go to this tree on this day at this time and I collect a few branches, I may keep the best one for my herbarium, but then I'll send these duplicates out to other herbaria around the world who may be interested in either that group of plant or maybe the location. They have a strong collection from Texas as well. So a lot of our Texas stuff we'll usually send to other herbaria across Texas and Oklahoma to strengthen their collections. Because again, I mentioned earlier, these are flammable. These can be destroyed. Anything can happen. So it's almost like an insurance. If I know that there's duplicates of this plant material elsewhere, if something happens to one, we know that at least it's safe, elsewhere and protected elsewhere. Yeah.
I'm very interested or impressed by these specimen that
what you got on yours?
You brought Mexican apple. Parts of my growing up was in the Philippines.
Oh, that's wonderful.
And the southern, in fact, just reminded me, I get goosebumps, reminded me that we have these trees right in our backyard.
Oh my gosh.
It's in the central Philippines. And we're on the coastal. We've been, collecting leaves since I was in high school, and just brought back a lot of memories. We still have houses there in the islands, and there's about 7,500 islands. 75, that means 7,500.
Yes.
Islands that are smaller islands. I don't know what your schedule is, Boracay, but I'm planning to go sometime
So
next year, and we could tour you with eagles. Gosh, and the list goes on.
So
Have you
I
been to
didn't.
Boracay? Is this your first trip to Philippines?
So I did not get to go on this trip. My boss actually did get to go on one of the expeditions. So our last expedition was last year, but our botanists are planning to resubmit a proposal to National Science Foundation to return to the Philippines. I just don't know. It'll probably be a different island, but I just don't know. They primarily were in Mindanao kind of area, so I'm not sure maybe what the next steps are, but I know they do want to return because there's a lot of work to do and a lot to explore. There's a lot of interesting plants over there.
Yeah, southern Mindanao is a little bit, I would just kind of caution you about the
They've
south.
had some interesting experience, but they work with local botanists there, and so they feel really good about kind of going out there, and they have all the permits they need and all that. Yeah.
Central, we go by Leyte. When General MacArthur came back to Leyte in summer during World War II, this is just like, part of my growing up, Hawaii and Spain. But the Philippines really first my love because of the trees, the natives, and even, this butterfly flutterby we had this weekend. It says here in the larval host plant for ruby spotted swallow butterfly, this one. So they might want to add, they call it Mexican apple, but this can be found in coconut groves. They're very much alike.
Yeah.
Because in fact, we have collected a bunch of leaves like this. So I was showing it to Jeff, and it just kind of gave me goosebumps.
It could be that you have like a closely related relative. But like I said, we had a volunteer kind of put all the information on the backs of these sheets. So who knows? He may have found like one common name and like stuck with it. But that's why we typically use scientific names to talk about plants with people from around the world, because there could be local or common names that are familiar to some people, but like not to others. But that's interesting. I'm so glad that you've seen something caught your eye that was familiar. Yeah.
I love books, and I'm going to brag. The Native Plant Society, we had all the authors on the very first North Central. I have the book that Rachel's first in line. Rachel's first in line. If I disappear, she gets my copy. And I think I've got the East Texas copy, but do y'all have a gift shop or book selling corner?
Yeah.
I haven't been to BRIT.
Yeah, you can. We do have an online shop for the BRIT Press, so you can order any of these books online. Well, there's like 2 of them that aren't sold through the BRIT Press. I just wanted to bring them. But yes, all of our floras are sold online, so you can get them.
You have to ship those heavy suckers?
Or if you wanted to make it a day trip, you can go to the garden, and if you go over to the BRIT building, we have a bunch of our books in that front atrium area, so you don't even have to walk far. Yeah. Yes. And some of them are even discounted if they're, there's some wear and tear, which really it's like a bump on the one side of it. So I know these heavy books can be expensive, but there are some discounted if they have, minor defects or something like that, which I usually go for. Yeah.
What about the marine plants?
Marine plants. Yeah. So we do have an algae collection. It's very small, but it's a little bit different than collecting with these field presses, right? Because you have to get them from underwater and they're wet, and how do they lay flat? So one of the techniques that I've heard that botanists will use when they're collecting algae is they'll slip some kind of paper or some kind of level underneath the water and basically bring it to the surface like this so it lays flat, rather than trying to like pick it up then lay it flat on the sheet, because it's hard to work with something when it's wet like that. So yeah, that's typically what they'll do. They'll like kind of bring it to the surface with a sheet of paper underneath, and usually that will be glued onto an archival sheet later on. I can bring up an example if you want. We just uploaded our macroalgae. So this is one of our online portals. So for each group of plants, they have a different portal. So this is for algae. And what I'm doing is I am searching, and down here are all of the herbaria with algae collections that have data on this portal. So I'm just gonna select ours, and then I'm gonna see algae from Texas. I'm gonna click search. And here you can see tiny icons of the images right here, and then some of the data starting to be over here. So if I click on right here, this was collected by Eula Whitehouse in 1947. So she looks like she originally put this in a packet, but this is quite a wad of algae right here. But this is like glued, it is almost like appended to this sheet that was eventually glued onto this larger sheet. Does anyone have a plant? I mean, I know I have a lot of plants out here.
Grapevine Lake had a big balloon.
Grapevine Lake?
Yeah, put that in there and see what comes up.
Oh, yeah. Oh, I don't know if we have algae from Grapevine Lake. Let's see. I can show you other specimens collected in Grapevine of our vascular plants. That I will find results for sure. So I'm gonna do Texas. I wonder if I put Grapevine in the locality information, and then I just want to see the BRIT collection. Check it out. And they may not be exactly our grapevine, depending on the county. There's a few other grapevines in here. But yes, here is a 1939 collection. All it says is near Grapevine, but let's look at it. Looks like a sunflower of some kind. So what's great is I can really zoom in and get a close-up of this plant itself, and then right down here in the corner, we've got the label data. So there probably wasn't much around here for him to give a description about where he was. You know, near Grapevine somewhere in some field. So we have this primary label, but I didn't mention that there are other labels that you might see on these specimens. These are called annotation labels. So typically it's not the collector, but a botanist later on that again might be studying this group Coreopsis, and you'll see some of the information on these labels. But they're basically either agreeing with the original collector that that is the identification, but in this case, they decided it is not Coreopsis drumondii, it is actually Coreopsis tinctoria. So it's important for botanists to continue looking at these specimens, annotating them, and making sure that they're even identified to the right thing. Because you could have something totally misidentified in a random cabinet somewhere, and it may not be discovered. And that's the point of putting this digitized data online, so more people, more botanists can actually see these images. And many botanists will just identify our stuff based on the images themselves. They'll send me an email and say, hey, you know, these specimens right here are not identified correctly, this is what they should be. And it's super helpful because I don't have all the plant knowledge in the world, so we're utilizing the community of botanists to help us make this a better collection, make these specimens better.
I have a question, please. Do they put, I want to know the difference. So how did guy B discover, no, you identified it wrong and it's based on this little thing that I saw here?
That's a really good question. So, you know, the herbarium is a big reference library. So I said earlier, I can go into one cabinet and look at one species from our state and see it across space and time in our state. And so that right there is already a good just visual. You may visually be able to see what is standing out. Does one of these not look like the other? So that is one way that botanists can kind of obviously tell if something's misidentified, but it's also through using floras. those floras have really accurate descriptions with, you know, the number of petals or the certain measurement of the leaves. So using the descriptions from floras also helps us with identifying specimens. And maybe these folks in 1939 didn't have those resources, but now that we do have these resources, we're able to accurately identify these specimens. And new species could be discovered as well. So a lot of people think that, oh, when you're out tromping in the field, that's when you find something new. But really, it's back in the herbarium, again, when you're able to kind of see everything in one spot and notice kind of any obvious differences or what's standing out. Even the location, is there a mountain in the middle of the range of this one species? And is one side a variety, or could one side of the mountain, those plants look a little bit different than the other side? It's totally possible. Thank you guys for letting me speak and share all the things about herbaria and what's going on. Thank you. Yeah, and I'm up here if anyone has anything else.